Welcome to Sumit. So today we learn about the very basic informations of zinc finger motif. So zinc finger motifs are actually that motifs or DNA binding motifs that will bind with zinc ions and form a finger-like structure. Okay. Now zinc ion is actually stabilizing the protein folding and it maintains the 3D structure. Okay. The most widely occurring zinc finger motif is C2H2 zinc finger. Here you can see I have written it here. C2H2, that means cysteine 2 histidine 2. Okay, but before that we have to know their functions. Their functions are like DNA recognition, RNA packaging, transcriptional activation, or protein-protein interaction, or you can say RNA binding, or you can say regulation of apoptosis, etc. Okay, so this is like CRISPR-Cas9. You know that in CRISPR-Cas9 it is used as genome editing technology, and zinc finger motif is also used in genome editing technology, but in a different way. Okay, now here coming to the third point, that C2H2 zinc finger was first discovered in TF3 alpha. Okay, sorry, TF3A, and each finger is actually 30 amino acid long. But before that we have to know the structure. Okay, zinc finger motif actually has two antiparallel beta sheets and followed by alpha helix. Here you can see this image for C2H2. For C2 means 2 cysteine and 2 histidine. Remember, the cysteines are in the antiparallel sheets and histidine are in the alpha helix. Here you can't understand in this diagram. Here I have tried this diagram, but you can't understand from these two. So let me try to draw it here. Let's say this is the antiparallel sheets. Okay, antiparallel beta sheets and followed by alpha helix. Okay, now here comes the zinc in the middle, and as I have said, two cysteines will be in the sheets, antiparallel beta sheets, and histidines are in the alpha helix. So zinc are bind with the two cysteine and 2 histidine. So that is like a finger-like structure. So this is why this is zinc finger motif of C2H2. Why? Because 2 cysteine, 2 histidine. Now it can be C2C2 also, or you can say C4. Okay, but before that, check that zinc is, there is one zinc, right? That's why it is called as monomeric. But for C2C2, there will be two zincs. That's why it can be called as dimeric. Okay, the mostly it will be seen in the steroid receptors. Okay, and the DNA binding domains of these factors will be of 70 to 80 amino acid long. Okay, and obviously will contain 2 zinc ions. That's why dimeric. So C4 or C2C2 is dimeric, C2H2 is monomeric. Okay, and both of the C2H2 and C2C2 will have their own particular consensus sequence. Okay, now this is the very basic information of zinc finger motif. I hope you have understood the very basics. So see you in the next video. Thank you.